Antibody structure and diversity. Antibodies or immunoglobulins are glycoprotein molecules which are designed to provide immunity or protection to host against infectious organisms. The antibody The antibody can recognize the proteins called antigens originating from such infections as non-self and in turn could neutralize the antigen and or the organism producing the antigen protein. Antibody constitute important component of humoral immunity. There are five types of antibodies characterized by heavy chains they possess: IgG, IgM, IgD, IgA, and IgE. An antibody response to an antigen generates polyclonal antibodies involving their production of a number of different types of immunoglobulin against the antigen. Each antibody is produced by a specific lymphocyte B cell. A single antibody species only IgM or IgG or IgA for IgD or IgE, only one sequence, therefore produced by a single lymphocyte clone, is called monoclonal antibodies. A polyclonal antibody response is more potent as it can bind to multiple epitopes on the antigen, neutralizing it more effectively. Polyclonal antibodies occur in maximum concentration in the serum. Immunoglobulins are polypeptide proteins and possess electrophoretic mobility. The overall charge on protein depends on the collective charge of the constituent amino acids and thus on its amino acid sequence. Various aspects of antibody. Various aspects of antibody would comprise production and natural distribution of antibodies, fine structure of antibodies, different classes of antibodies. इनको हम parts में करेंगे क्योंकि बहुत लंबा हो जाएगा नहीं तो. Production and natural distribution of antibodies. Out of all somatic cells, only B cell produces antibodies. The B cells are the only cells in which the blueprint of original DNA changes and rearranges to produce antibodies. These antibodies are distributed throughout the body and are also found on the surface of B cells. Antibodies are also found in endoplasmic reticulum and Golgi complexes. The surface antibody of B cell encounters with antigen and B cell is transferred into plasmocyte which secretes specific antibodies against specific antigen. The secreted antibodies are often attached to the surface of neutrophil, mast cells, natural killer cells which possess specific receptor for binding antibodies. During clot formation in blood, the antibodies remain in residual fluid and they bind with antigen and such serum is called antiserum. The serum antibodies are quantitatively determined by radioimmunodiffusion and by enzyme-linked immunosorbent assay ELISA technique. Fine structure of antibody gamma globulin IgG. The fine structure of IgG has been described as per H2L2 model proposed by Rodney Porter and Gerald Edelman. IgG comprises classes and kinds of light chain, classes and kinds of heavy chains, folding domains, various constant and hypervariable regions, globular domains and beta plateacids, and secondary structure of antibodies. IgG is observed as Y-shaped structure. The two arms of the Y serve as two antibody binding site for antigen. Each antibody site is located on the one of the arm of Y between a heavy chain and a light chain. These are called fragment AB region. Tail of Y form a third region called FC region. Both light and heavy chain possesses variable and constant regions. Light and heavy chains. Light and heavy chains consist of different classes of polypeptide chain. Classes of kinds of light chain. Each immunoglobulin contains two kinds of light chains and two kinds of irrespective of its antigen binding ability. These are called kappa and lambda chains. An antibody or immunoglobulin contains either kappa or either lambda chains, light chains, never both. The ratio of kappa or lambda chain varies between different species or in the same species. In man, 65% antibody possess kappa chain and 35% contains possess lambda chain. They vary in their amino acids. The kappa light chain polypeptides are coded by DNA segments of chromosome 2 in men and 6 in mice. The lambda light chain polypeptide are coded by DNA segment found in chromosome 22 in men and 16 in mice. Classes of kinds of heavy chains. Heavy chains have been differentiated into following classes: gamma for IgG, mu for IgM, alpha for IgA, delta for IgD, epsilon for IgE. The two heavy chains of antibody are joined together by interchain disulfide bonds at the site called hinge region. The light and heavy chains are also joined together by interchain disulfide bridge bonds. The heavy chain polypeptides are coded by DNA segments on chromosome 14 in man and 12 in mice. In case of man, the heavy chain of gamma chain of IgG is further classified into subclasses such as gamma 1, gamma 2, gamma 3 and gamma 4 for IgG1, IgG2, IgG3 and IgG4 antibodies respectively. In IgG1 and IgG4, the heavy chain at hinge region are joined together by two interchain disulfide bond. IgG2 has 4 interchain disulfide bond in its heavy chain called hinge region. The Ig3 hinge region possesses 7 disulfide bond. Ig3 has shortest half-life. Ig3 also possesses lowest synthetic rate and highest catabolic rate. Folding domains and variable constant and hypervariable region. The polypeptide chain of immunoglobulins or antibodies reveals folding domains and variation in amino acid sequence in variable and hypervariable regions towards N-terminal. Light chain polypeptide contains about 214 amino acid residue in its 2 domain, while heavy chain has about 445 amino acid in its 4 domain. The region in the heavy chain towards COOH terminus is called constant heavy chain region. The constant heavy chain region domain possesses 3 constant regions: CH1, CH2 and CH3. In the variable region of both light and heavy chains, the variation in amino acid sequence is largely restricted to 3 smaller regions. These smaller regions with about 10 amino acids each are called hypervariable region. The hypervariable region serves as contact site for antibody binding to antigen. These regions are called as complementarity determining region or CDRs. The CDRs chain in main constant consist of CDR1 having 24 to 34 amino acid residue, CDR2 with 50 to 56 amino acid residue, CDR3 containing 89 to 97 amino acid residues. Between CDR1, 2 and 3, there are regions where amino acid sequences are relatively constant. Such regions are called framework region FRCs. Thus, framework region in light chains are FR1 with 1 to 23, FR2 having 35 to 59, FR3 with 57 to 88, and FR4 containing 98 to 107 amino acid residue. In case of heavy chain, the amino acid residue consists of ये हो गया. In case of heavy chain, the amino acid residue consists of 31 to 35 for CDR1, 50 to 65 to CDR2, 96 to 102 CDR3 respectively. It has been further observed that the amino acid sequence in constant region are not absolutely constant. They show some variation in amino acid sequences. In framework region, variable region have been classified in four subgroups of V kappa region, 6 subgroup of V lambda, and 4 subgroups in V heavy region. In antibody binding region, the place of attachment of antigen with antibody is called epitope, where motif of antibody attaching with antigen is called paratope. The CDRs of both heavy and light chains take part in antigen-antibody binding. The light and heavy chains are folded in such a way that the three complementarity determining regions in each chain constitute wall of antigen binding group. Biologically, IgG molecule shows two antigen antibody binding site in variable region of light and heavy chain, complement activating site in CH2 region of heavy chain, and other binding site as CH4 region of heavy chain. The light chain contains one globular domain in variable region and one constant domain in constant region. Each heavy chain contains one globular region in variable region and 3 to 4 region in constant region, CH1, CH2, CH3, CH4 depending on the class of antibodies. In IgM and IgG antibody, the constant region contain 4 globular domains such as CH1, CH2, CH3, CH4 in tandem arrangement, whereas C region of IgG, IgD and IgG antibodies possess 3 globular domains such as CH1, CH2, CH3 in tandem arrangement. All the globular domain of light and heavy chain contain several homologous unit of about 100 amino acid residue within each folded domain. Within each folded domain, interchain disulfide bond involving about 60 amino acid residue which form a loop. The pattern of folding of complementarity determining region constitute wall of antigen antibody binding group. Globular domains and beta-pleated sheets immunoglobulin are made up of primary, secondary, tertiary and quaternary structure of proteins. The VL domain is longer, consisting of 16 amino acids. There are 5 light grey color antiparallel flat arrows and 4 dark color antiparallel arrows. The dark black colored bars and interchain disulfide bonds in each sheet. Constant light and variable light domains are found in the extracellular region of the many membrane.